Okay, great. Welcome everyone to the Precision Medicine Seminar. It's my pleasure to introduce a guest speaker today, Soumya Raychaudhuri from the Brigham and Women's Hospital in Boston. Soumya is a professor of medicine at Harvard. He's also a professor of biomedical informatics at the Harvard Medical School. He directs the Center for Data Sciences at Harvard, and he's also an associate member of the Broad Institute. He's clinically active as a rheumatologist and sees patients at the Brigham and Women's Arthritis Clinic. He completed an MD PhD program at Stanford University, and then he pursued a clinical training in internal medicine and subspecialty training in rheumatology at Brigham and Women's Hospital at Harvard. And he also, concurrently with his subspecialty training, completed a postdoctoral fellowship training in genetics at the Broad Institute with Mark Daly. And since joining the faculty at Harvard, he has been contributing to our understanding of the genetic factors in rheumatoid arthritis and also other immune-mediated diseases. He has been really at the forefront of devising different methods to localize genetic signals, genetic associations to causal variants, causal genes, and integrating genetic information or functional information to better understand the genetic mechanisms. He's the recipient of multiple awards, including Doris Duke Award, Young Investigator Award from the American College of Rheumatology. He's elected member of the American Society of Clinical Investigation, and he has multiple active research projects right now in human genetics and functional genomics of rheumatoid arthritis, and a specific focus now on genomic strategies to understand CD4 plus T-cell biology. So it's my pleasure to welcome Soumya to the seminar, and we're really looking forward to learn about your latest work.
Okay, well, thank you, Christoph, for the invitation and for the really nice introduction. So I'll be talking about using single-cell genomics to understand the basis of tissue inflammation in rheumatoid arthritis today. And my lab really is focused on trying to understand autoimmune diseases. As all of you will know, autoimmune diseases are a spectrum of medical conditions that can affect an individual organ. It can be highly organ specific, but also can be systemic. The particular condition I think the most about is rheumatoid arthritis. Rheumatoid arthritis is an autoimmune disease where the immune system infiltrates the synovial tissue of the joint. And this is Pierre-Auguste Renoir, the famous French painter. And you can see that when he painted this self-portrait, his hand was so damaged by rheumatoid arthritis that he was no longer holding a brush in his hand. So paintings like this were done with a brush strapped to his wrist. Therapies for RA have really improved dramatically in the past 3 decades, but there's still gaps. Still a lot of patients don't obtain remission. There's a need to identify the right treatment quickly. The slower you are to treat, the more problematic the long-term damages. And then there's a need for novel therapeutics, since most of our treatments don't consistently lead to remission, and there's still no cure. And of course, in RA, the joint is inflamed, and it's this particular membrane, the synovial membrane, which surrounds the joint capsule, that becomes infiltrated with immune cells, and that is what leads to joint destruction. Since 2006, I've been involved in genetic studies of RA. There's some folks have been involved in the genetic study of RA much longer than that here in Columbia University. One of the things we've found is that a lot of disease alleles implicate T-cell biology. Of course, there's the very important association inside the HLA and HLA-DRB1, which implicates antigen presentation, T-cell interactions, and then the other risk alleles that have emerged, the rest of the genome, also seem to implicate T-cell gene regulation, and I'll talk about that toward the end of the talk. So genetics is one way to get at the mechanism, but another strategy is, can we simply define the key T cell states or key cell states in tissue inflammation? So one key question is, what are the driving relevant cell types in RA? So I'm going to talk about, in this talk today, I'm going to spend most of my time on the first part, which is talking about identification of key cell states in the context of rheumatoid arthritis tissue inflammation. This is work done by myself and many others as part of the AMP consortium. And the latter part, I'm going to talk about genetics and how we've been using that to understand gene regulation in key cell states. And a lot of what I'm going to talk about today is going to have a lot to do with single cell data. And obviously, it will escape no one's attention that over the last decade, we've gone from being able to analyze only a handful of cells in the earliest experiments to being able to routinely look at thousands and tens of thousands of single cells to generate whole transcriptome and increasingly whole multimodal single cell data. And in terms of the tissue work, I'm going to focus on work I've been doing in conjunction with the Accelerating Medical Partnerships program. This is a joint public-private venture to understand rheumatoid arthritis. There's also a lupus wing to it as well. There are two phases to this study. Phase one, where the goal was really to collect data on single cells for synovial joint biopsies. The goal was to define key populations and to identify and characterize the populations that are expanded with inflammation. Phase two, it's really to understand the heterogeneity in RA patients and starting to think about what the different types of RA are and whether there's a path to precision therapeutics. And without going any further, I just want to acknowledge the breadth and comprehensive expertise in the AMP consortium. As you can see, it includes many primary network sites, many secondary network sites. I'm privileged on this call to have several of the co-authors on the latest AMP RA manuscript with me. I want to especially acknowledge Michael Brenner, Jan Analik, and Laura Donlin, who are my co-leads on the RA study. And in the beginning, first I'm going to talk about the phase one study, which is that smaller initial study, and that was work that was co-led by Fan Zhang, now in Colorado as an independent faculty member, Kevin Wei, Camille Slowakowski, Chama Fonseca, and Deepak Rao. And all this data is in the public domain and can be easily downloaded or queried online. So in this study, the goal was really to define the key cell states that were relevant to RA. So the idea was that we had clinical sites where we obtained synovial tissue here from the joint, and those synovial tissues were sent for multiple assays. First, of course, we wanted to see the histology. Then we disaggregated the cells and looked at mass cytometry and flow cytometry to get single cell data. And then we sorted the cells and applied both bulk and single cell RNA-seq so we could characterize the key pathogenic populations in RA. So there were, in this initial study, there were 4 data types that we were looking at: flow cytometry, bulk RNA-seq, mass cytometry, and single cell RNA-seq. And we had patients that had OA as controls. We also had 2 groups of RA, leukocyte-rich RA. These are highly inflamed RA patients, and what we call leukocyte-poor RA, which look more kind of like burnt out RA or RA where the inflammation is no longer obviously present. So these individuals looked a lot like osteoarthritis. We had a total of 51 patients, and before I get into the main results, I just thought I would quickly highlight the big findings of this study. So in this study, we were able to define 18 unique cell states. We've since expanded on that dramatically. Single cell and RNA-seq, single cell RNA-seq and mass cytometry data together showed a dramatic expansion of inflammatory fibroblasts, 34-fold between RA and controls. IL-1B positive pro-inflammatory monocytes or macrophages were identified as 8-fold expanded. Autoimmune-associated B cells, a helper B cell population was 6-fold expanded. T peripheral helper cells which are similar to T follicular helper cells, but seen in tissue, were 3-fold expanded. And we were able to identify the cellular sources for a key cytokine. So for example, IL-6 largely came from inflammatory fibroblasts, Thy1 positive HLA high fibroblasts, and IL-1 largely came from pro-inflammatory monocytes. So in this initial study, we had 5,000 cells, which in 2018 seemed like a lot of cells. We were able to identify 18 main populations. And I'll give you a little bit of a flavor of how we analyzed the data, how we thought about the data by some of the work that we did here in the fibroblasts. So you can see here that we had something like 1,000 fibroblast single cells across about 18 samples. We clustered them and we found that there were 4 major fibroblast populations. SCF1, SCF2, SCF3 were sublining populations deep in the synovium. And then there was SCF4, which was a lining population. And in the middle here, you can see the single cell embedding. And what you see right away is IL-6 and HLA are expressed by that SCF2 population. That's the HLA high sublining population, a population of fibroblasts that we thought was pro-inflammatory. Now remember, we had very limited single cell data, so we were able to use the single cell data to say, what's present and what's not present across many samples. But to really understand what was upregulated or not, what we needed to do was we needed to take the marker genes from the fibroblast data, as we've done here, and then look at differential expression of those marker genes. And what we found is that the marker genes for those HLA high fibroblasts were indeed expanded in our RA cases compared to the controls in a dramatic way. We weren't satisfied to look at one technology, so we also had access to mass cytometry data. This is the mass cytometry data that you see here. This is similar to flow data, but with many surface markers. And in this case, we again defined key populations, and you see this Thy1 positive HLA high yellow population of fibroblasts right over here. And now when you look at our samples, you see that the leukocyte-rich, the inflamed RA patients have this big yellow presence. We apply a statistical technique called MASK. And what we're able to see is that there's a dramatic expansion, about 34-fold expansion of this HLA high fibroblast population in RA. So the landscape of fibroblasts is dramatically transformed in RA. Where are these cells? So this is fibroblast tissues that we're looking at. On the left, you're looking at PDPN positive staining, which tells you where the fibroblasts are. On the right, you're looking at CD45 in blue. That tells you where the immune cells or the hematopoietic cells are. And now we stain for Thy1 positive. That's pointing to that big group of inflammatory fibroblasts. A subset of these are HLA positive. And look at that green ring. You can see it's dramatically expanded around the blood vessels. And that's a much thinner population in OA. Kevin Way, who was one of our key colleagues, went back with Ilya Korsunsky, who was then in my lab. Now they're both now faculty members. And they took a look at these populations. And what they saw is that there was a continuous gradient between sublining and lining. And that this population we saw expanded kind of represented an extreme. And what they found out is what was happening was that the vascular endothelial cells were engaged in notch signaling of the populations around them, and they were transforming the fibroblast populations. Sometimes these results, and we'll talk more about how to use single cell data to think about genetics later on, but some of these results can be really useful. So this is again, our 4 fibroblast populations. Look at this lining layer. If you look at the marker genes, you see that there's this one population, an important gene for RA and for lupus. It's very specifically expressed in the lining layer. Now look at this. On the right, you see the DNase locus and the genetic association of SNPs in that locus to RA. And you find that there's this one SNP that's very strongly associated with RA susceptibility. It's a loss of function. It's an arginine to cysteine change, which abrogates DNase 1L3 function, results in RA. It's very specifically expressed in this one synovial fibroblast population, giving us a clue as to how we can use single cell data to understand how disease genes are working. So to summarize the fibroblast results, we found 4 key populations, including this SCF2 inflammatory fibroblast population, which has expanded in synovial tissues of RA dramatically. I showed you evidence from bulk RNA-seq, mass cytometry data. This population specifically secreted IL-6, and it's an inflammatory population driven by Notch signaling. There's a lining population, which is actually depleted in RA, which expresses a genetically implicated gene, DNase 1L3. So I'm not going to get into all of the other populations that got us to the other T cell types. I'm simply going to point out that we use the same process to find these other populations that are driving, that are expanded in RA inflammation in the synovium. So the phase 1 study that I told you about implicated T peripheral helper cells, age-associated B cells, inflammatory fibroblasts, which we talked about extensively, and IL-1B positive inflammatory monocytes. But we did not assay enough RA patients to understand how these cell states correlate with each other. So is it the case that if you have one of these populations, you have them all, or is there diversity? Are there some patients that have a preponderance of inflammatory monocytes, and maybe they should be treated with anti-IL-1? Are there other patients who have a ton of inflammatory fibroblasts? Maybe they should be treated with anti-IL-6. So there are 2 possibilities, of course, that all forms of RA are the same. A second possibility is that different forms of RA are different given the different pathogenic cell types. And of course, this has implications for precision therapeutics. So to get at this, we really need a larger study. And that's where phase 2, we all teamed up again to do a phase 2 study, which is a larger study to characterize heterogeneity in RA patients. Without getting into all of the major differences, technology had passed, time had passed, technology had increased. So you can see in phase 1, we had 5,000 cells. Phase 2, we had 300,000 single cells. Phase 1, we had plate-based single cell sorting. Phase 2, now we're doing droplets, so we're able to get a ton more data. Phase 2, also importantly, we have integrated RNA and surface marker data. And then we have a bunch of other ancillary data, such as ataxia, T cell repertoire, et cetera. Phase 1, the goal was, can we define those pathogenic populations, pathogenic cell states? Phase 2, can we define cell populations and how they covariate together? Can we understand clinical heterogeneity? And I won't get into all the different facets of the data, but suffice it to say, we had a wide spectrum of data types. Also, I just want to thank all the folks involved in analyzing the data because of the increased complexity. You can see we had a pretty big team of people who were working on making sense of the data. And in phase 1, we were using plate-based techniques in 2014, 2017, 2018, when we were collecting most of this data. We were able to generate 10,000 or more single cells per sample. So here's the pipeline run by Kevin Way and Helena Johnson and Michael Brenner at Boston. The synovial biopsies came from many sites, including many from Columbia University, where the samples were obtained, histology was done, disaggregation happened. And again, those cells were sent, this time through droplet-based sequencing to generate single-cell RNA-seq data and site-seq data. We wanted to capture the full diversity of RA patients. We didn't want to look at a homogeneous group. We wanted to see the spectrum of people with RA. So we had individuals who were DMARD naive. They had not been put on an immunomodulatory yet. They were effectively newly diagnosed RA. We had individuals who were treated with first-line therapy, methotrexate, but were inadequate responders. They were likely to need a stronger immunomodulatory like a biologic. And we had individuals who had been put on the main biologic anti-TNFs, but had not responded. So we had individuals with sort of different degrees of disease. Here are 300,000 cells, and again, you can see we're able to first simply call these major cell types, NK cells, T cells, myeloid cells, etc. And then what we did was we took our patient samples and we looked at, how many T cells do they have, how many fibroblasts do they have, how many myeloid cells do they have, and then we're able to cluster them. And what we found is that there were 6 groups of RA. We called them EFM for those individuals who have lots of endothelial cells, fibroblasts, and myeloid cells. F for the that have a lot of fibroblast cells. T plus F for those individuals that have a lot of T cells and fibroblasts. T plus B for those individuals who have T cells and B cells and so on. So we had these 6 groups, and on the right you see the principal component plot, and basically what you see is it's really a continuum. On one end of the spectrum you have EFM, which is similar to F, which is lots of fibroblasts, lots of stromal populations. In the middle there you have T plus B, individuals that have lots of T cells and B cells, and then finally you have inflammatory, finally have macrophage enriched individuals. So when we looked at what we found very interestingly is that the CTAPs tracked with histology and serologies. They weren't really different across the 3 different clinical cohorts, but what we saw is individuals who had, when we looked at the histology and we scored the histology for density, which is how much immune cellular infiltrate there is, or aggregate, which tells you how many kind of lymphocyte aggregates there are in a sample. You see that these CTAP groups are very different across these scores. You also see that the main antibody for RA, CCP antibody, it's diagnostic for RA with 70% sensitivity. The titer of CCP vary dramatically with T plus B RA, which is maybe the most adaptive immune type of RA, having the highest CCP titer. But it doesn't track with disease activity. One of the things we thought is maybe T plus B RA would have the highest disease activity. No, if you look at DAS28 CDAI, common metrics of disease activity, actually that's not statistically different. So taking a deeper look at the single cell data, what you see is that there's 77 different cell states. And for each of these populations, for T cells, for example, you can see that there's roughly 20 different cell states that we were able to define. So how do we think of this in the context of these CTAPs? So here we have myeloid cells. You can see that there's a whole spectrum, of them here. We've annotated them as best as we could. This includes dendritic cells. You can see that M7 is those IL-1 positive, M6, M7. That's these guys in the middle. Those are those inflammatory populations. So which populations are associated with specific CTAPs? So we did a simple analysis. We looked at the proportion of each cluster in a myeloid cell and simply asked, do they have more than average, a more than average percentage of their myeloid cells are within a specific cluster? And then we can define a Z-score to give us a statistic. And you start to see patterns like this, that some populations are overrepresented and underrepresented. So for example, if you look at M and T plus M, you see that M6, M4, and M3 have red boxes suggesting that those populations are enriched. And indeed, you can do this for all of the different single cell populations. And you start to see that there's kind of a complicated pattern that's a little bit hard to make sense of, but there are a lot of differences. And here's another example. When you look at the T plus F or T plus B RA, you see that there's T cell populations that are enriched. Both of these types of RA has a lot of T cells, but they're different T cell populations that you're seeing that are enriched. So to get a better picture of this, we needed new statistics. So we realized that clustering and defining populations may not be ideal. So we defined this strategy called covariant neighborhood analysis. So clustering forces us investigators to make arbitrary choices about how big or how small, where the boundaries are between clusters. And transcriptional heterogeneity might be more of a continuum. So our approach was to define local neighborhoods of cells and use correlation across the samples to define relevant groups of neighborhoods. So this is a little bit of a sense of how we do this. This is work by Yakir Reshef and Lori Rumker. What we do is we start with an individual cell in our cell space, and we identify the cells that are most similar to it, and we do a random walk to define a neighborhood. Now you can see we have 5 different neighborhoods here, A, B, C, and D, and we can go and look at our samples and say, how well represented is each patient in the neighborhood? And we can define a matrix called the neighborhood abundance matrix. So you can see for neighborhood A over here, it has a lot of cells from patient number 3, not so many cells from the other patients, maybe a few cells from patient number 4. So that gives you a profile. Now you can apply standard techniques like principal components analysis to kind of group cells together that are moving similarly across patients. And now you can do an association test and identify the neighborhoods that are driving a phenotype. So these are the NK cells. Again, you can go through and do what we were doing, and you can see that there are clearly some populations like NK0123 that are enriched here. So CD16 population seems to be enriched in T plus FRA. But now you can apply covariant neighborhood analysis, and now you get a very clear picture that there's this one set of cells which represents multiple clusters, which are all expanded in T plus FRA. And that happens to be the interferon gamma secreting NK cells. So they're depleted in some forms of RA, but in T plus FRA, this is expanded, and these are the interferon gamma secreting NK cells. What about more broadly? What about T cells? Now you look here at the left and the right, TB and TF, they're both RA samples that have a lot of T cells, but look at that, they have different T cells that are represented. On the left, you see T3, T7. These are CD4 T cells. They're adaptive T cells. They're T peripheral helper cells. On the right, you see more of the kind of CD8 population, T12, T15, cytotoxic T cells, CD4 and CD8 T cells, mostly secreting interferon gamma. So T plus FRA has a different set of T cells that are driving that type of RA than T plus B. What about B cells? Another example here. When you apply CNA, you see that your memory B cells and your age-associated B cells, remember those age-associated B cells from phase 1, they're really enriched in T plus B RA. And myeloid cell, very interesting story. Whether you look at E plus EFM, those are the endothelial fibroblast and myeloid cell RA, versus fibroblast RA versus myeloid cell RA, you see that there's different groups of macrophage and myeloid populations that are enriched. In particular, if you look on the right, you see that these these cells in the middle, which represent your inflammatory macrophages, STAT1 positive, CXCL10 positive, SPP1 positive, those are the enriched populations. So different types of macrophages, different types of myeloid cells, depending on which kind of RA you have. So CTAPs have, they favor different cell states, as I think I hopefully convinced you. T plus F, for example, favors interferon gamma positive NK cells, interferon gamma positive T cells. T plus B favors T peripheral helper cells or age-associated B cells. So we can now make a whole map of these different CTAPs, and we can show which populations are playing a role, are expanded or depleted in each of these flavors of tissue inflammation. So if you look, for example, at FRA, so the types of samples that have a lot of fibroblasts, well, they have a lot of different things going on, including in the endothelial cells, where venous endothelial cells appear to be expanded, arterial and endothelial cells are relatively depleted, and of course the other associations that I already told you about. So to summarize, we obtained data on 82 patient synovial samples and applied single cell technologies. We got data on 300,000 cells, and we were able to define 6 patterns of tissue inflammation based on the dominant cell type. Each cell type abundant phenotype has key cell states. And the examples, I showed you some examples of key myeloid and NK populations. And some of these populations were enriched or depleted in individual flavors of RA, and that has possible implications for therapeutics. And then to summarize on this whole section, first I told you about phase 1, where we identified key cell states associated with synovial inflammation of rheumatoid arthritis. There was a surprising cellular heterogeneity in the context of tissue inflammation. We were able to attribute cytokines to specific populations, and I told you about some key populations. And these populations are giving us key insights. And in phase 2, we were able to look at lots of patients all together, and we were able to see these 6 broad patterns of inflammation implicating specific cell populations. And also along the way, I made the suggestion that some key cell populations might play an important role in causing disease. You'd obviously be interested to know which genes are there. Are they implicated by genetics? Are they implicated by gene regulatory networks? And it's really important for us to understand the effect of risk alleles in individual disease-relevant cell types. And we talked about these key populations, but now we need to think a little bit about how genetic variation might be influencing cellular function. So I'm going to transition now a little bit and start talking about genetics. Rheumatoid arthritis has been a field that's been extremely successful in terms of genetics. This is the 2014 study led by Yukinori Okada and my colleagues Kazuhiki Yamamoto and Robert Plenge. This identified 100 risk alleles for RA. There's now over 150 risk alleles for RA that are floating out there. T cell biology, as I mentioned, is strongly implicated, including the HLA locus. And in the HLA locus, we think presentation of antigen to T cell receptor is really important. Outside of the HLA, heritability studies and fine mapping are pointing to T cell regulatory elements. And of course, many of you know that the HLA is important for antigen presentation to T cells. On the left here, you see a little schematic where you have an antigen presenting cell presenting with the HLA over here, presenting an antigenic peptide, causing T cell reactivity on the right. And that this is maybe causing a naive T cell to take on all kinds of different cell states. And of course, the key question is which of these cell states are important for genetics? Which of them are we finding in these tissue studies that we're doing? We and many others have known for a while, this is a study from our group, that if you take all these risk alleles and you say which histone marks are they enriched in, which open chromatin regions are they enriched in, they tend to be enriched in open chromatin regions that are important for CD4 T cell subsets. Now, you can't tell which one very easily, but you can see that there's an enrichment there. Similar study done by Kyle Farr, Mark Daly, and others, as listed here, that for autoimmune diseases in general, you see enrichment of T cell regulatory elements. So where does that leave us? Many risk alleles confer risk for autoimmune diseases within T cell regulatory regions. T cells can take on functional states, naive versus memory, naive versus effector. Effector populations can be very specific, Th17, Th2, Th1, mixed cell states. And the regulatory elements that might matter for genetic susceptibility might be active within specific T cell states. So understanding how these alleles work, which cell states they work in, and what genes they act on is essential for us to understand disease mechanism and to develop drug targets. So, and we and many people have been working on this problem, trying to annotate regulatory regions, especially in T cells. We think we've gotten a better ability to define regulatory regions based on open chromatin assays, et cetera. And we can look at things like expression QTLs, protein QTLs, cellular QTLs. So I present this example here of the CD28 CTLA4 locus, 2 really important genes for T cell biology, to give you a sense of a key challenge in defining functional alleles. So as many of you will know, when we do a SNP association study, we find a whole bunch of SNPs within a locus that are associated. Many of them are correlated with each other. So, that makes it hard to define which SNP is actually driving a signal. So this is linkage disequilibrium. Many disease alleles localized enhancers that have cell context specificity. And the challenge is a lot of times these SNPs aren't associated with expression. You would expect them to be, but they aren't. So here's an example of the CD28 CTLA4 locus. You see this these SNPs here. We apply fine mapping down at the bottom, and we think we have a SNP which is most likely to be causal. We did some luciferase assays, and we showed that the SNP is associated with enhancer activity and the C allele has more enhancer activity than the A allele, so suggesting that this allele is functional. It's in a regulatory region, which is really compelling, but it's not an EQTL. So that's the challenge. So our assumption is that a SNP like this is probably active in some key T cell states, but not in general active everywhere. So this is work by Aparna Nathan. So to get at this, we really wanted to come up with a strategy to define context-specific gene regulation in T cells. We wanted to profile memory T cells from 250 individuals. We used a Peruvian cohort, and we looked at 500,000 memory T cells and obtained RNA and surface marker data. For T cells, we really like single cell RNA-seq data, but we need to look at surface marker data too. When we phenotype T cells, RNA gives us a view of the heterogeneity. RNA is great to tell us which T cells are cytotoxic. It's great to tell us which T cells are proliferating. But if we really want to get at T cell phenotypes, we need not just global RNA markers, but traditional surface markers like CD25, CD62L, which demarcate effector versus central memory T cells, demarcate naive versus memory T cells, regulatory T cells, et cetera. When we started this study, we had access to CiteSeq, or one of these technologies where you have antibodies that you can label with an oligonucleotide, as illustrated here. And then when you do RNA-seq, what you do is you stain your cells with these antibodies, and then you sequence both the oligonucleotide of the antibody that's stuck to the cell alongside with the RNA. So you get a complete picture of both the RNA and the surface marker protein. Specific T cell panel, which we thought would capture the spectrum of T cell states. And then it became important to think about how do we think about RNA and protein together, because it's a massive, you have data now on thousands of genes and 30 surface markers. So we started with our samples, PBMCs, sorted T cells. We stained with those antibodies with the oligonucleotide labels, and we then obtained RNA and antibody data. And once we had the data, what we applied, it was a technique called canonical correlation analysis. It was produced in 1936, but we're using it here. And then the idea is that you're identifying combinations of genes that correlate with combinations of proteins. So you're finding linear combinations of genes and linear combinations of proteins to maximize the correlation between cells. And those are your canonical covariates. And they help you define populations of cells where the phenotype is reflected both on the protein side as well as the RNA side. So you see here clear separation between CD4 and CD8, a clear regulatory T cell CD25 positive population, and so on. And the canonical correlates actually give us a continuous variable that helps us functionalize the T cell. So the first one is cytotoxicity. So the cells on the right are more cytotoxic than the cells on the left. CV2 gives us a sense of how regulatory a T cell is. So cells at the bottom are regulatory T cells. CV3 separates naive and memory T cells, and so on. We argue that each of these CVs pick up on a specific function of T cells. And now we can use these variables to simply ask the question, are there EQTLs that are, not present on the left? So in cluster 14, you have a SNP, which is not associated with expression, but the same SNP in cluster 28 has a dramatic effect on expression. We modeled all our data using these canonical cell states, so using a continuous variable. And we found some very exciting results, and here's an example. This is the gene called MAF. You can see on the far left where things are very lightly colored, that there's really no correlation between the SNP genotype and expression. But as you make your way to the right, to those cytotoxic T cells on the far right, you see a strong correlation between the SNP and expression. Here's a really interesting gene, MDGA1. This has two SNPs that affect expression. The first one, the effect gets bigger as your T cells get more cytotoxic as you move to the right. And the bottom one, the effect gets weaker. This is the second SNP, that interior SNP. So you have two SNPs that are affecting regulatory control, but the cell state is affecting them in different and opposing ways. CTLA4, that SNP I told you about in the beginning, that locus I told you about in the beginning, one of the SNPs in that gene illustrated here associated with RA and type 1 is sometimes an EQTL and sometimes not. And in the cells in which CTLA4 is most highly expressed, it's not an EQTL. It doesn't associate with expression. And the ones where it's lowly expressed in T cells that aren't regulatory T cells, that's where you see the association. Here's another gene, RMDL3. So these state-dependent EQTLs include risk variants. IL-18R1 is associated with inflammatory bowel disease. And indeed, as we've systematically gone through this, we have found that 70% of the RA risk alleles are altered by cell state. And maybe in the interest of time, I will skip this. So to summarize, gene regulation can be dynamic across T cell states. T cell activation alters how SNPs regulate genes. Allele-specific expression or EQTLs change dynamically as you move from naive to memory to stimulated T cell states. Single cell EQTL studies, our study and probably others coming out reveal a broad range of T cell states. These cell states can be modeled with a continuous variable, and about a third of these EQTLs are dynamic across ex vivo cell states. And autoimmune disease EQTLs are disproportionately dynamic. So let me just conclude on this talk and have a few minutes for questions. So RA is a disease of synovial joint tissue inflammation. Genetics and single cell technologies complement each other and help point to mechanism in the human disease context. Single cell technologies are identifying key cell states and tissues. Here we demonstrate how synovial tissue cell states include pathogenic, synovial tissue samples include pathogenic cell states. RA synovial tissues demonstrate very different types of inflammation, favoring different inflammatory cell states. Cell state heterogeneity across patients exists and may have important therapeutic implications. RA genetics implicate T cells predominantly and T cell gene regulation. And there's a lot of interest in understanding how disease alleles alter gene regulation and cell function within these cell states. Many regulatory alleles, including disease-driving alleles, are dependent on T cell context. We demonstrated how regulatory effects can be disease-associated and enrich and enhance regulatory regions. So just as a final note, we're obviously thinking about future directions. We've identified how regulatory alleles are dependent on cell states. We've identified key pathogenic cell states. We want to put these things together. So we're very interested in trying to understand how do RA risk alleles act in disease-specific cell states? How does an allele act in, say, a T peripheral helper cell or an inflammatory fibroblast or IL-1 macrophage? Linking RA risk alleles to pathogenic cell states in RA will point us to causal disease mechanisms. They require us to define the regulatory effects of alleles within synovial inflammatory tissue. So I've thanked many people along the way. A lot of these studies involve a huge amount of effort from colleagues and collaborators from around the world. And I also want to just take a moment to thank my lab as depicted here. And then I'll take a few minutes for questions. Thank you.
Thank you so much. This was really wonderful and really impressive data sets. The session is open to questions. You feel free to post your questions in the chat, or you could possibly raise your hand, and I'm happy to unmute you. Okay, we have a first questions. Please go ahead.
Is it me, I guess? Hi, I'm, as somebody who does statistics, I find this whole field very, very new and challenging, and it was very helpful to hear the talk. So I have a really, really low-level question. At the end of phase 2, you were talking about like 80 samples and having like hundreds of thousands of, or maybe tens of thousands of cells that you worked with, and you came to certain conclusions. So I was curious, that seemed like a lot of samples to me compared to some other reports I've heard. What if you had divided the 80 samples into 2 separate groups and explored each one separately would you have come up with the same, conclusions as you did with looking at all 80 together? Thank you.
It's a great question. I mean, it's not something we did. I guess if we did it now, we would be cheating because we've now it would be a great thought experiment though.
No, but I think that you obviously followed certain procedure. So what if you applied that same logical process to a randomly divided half sample, and might be interesting. Thank you.
I think the answer is probably what we would see. So, you saw that, when we clustered on the major cell types, we found 6 basically groups of RA. My guess is with half the samples, we wouldn't find 6 groups. We'd probably find 3 or 4. I mean, again, they represent a continuum, really. So I think probably some of those groups would kind of glom together. I think we would have probably found some of the major findings, but we would have lost some of the granularity. I think there's no question about that.
I just feel that might really strengthen your conclusions. Some sort of sensitivity analysis. Yeah. Thank you. Thank you again.
Thank you.
So I have a related question, just sticking to the sample size question. So obviously single cell profiling is expensive. And in order to do EQTL analyses that are well powered, you need a relatively large data set like the one you described with 200 samples. What is in your mind an optimal data set or minimum data set for these type of analyses? And related question also is the data set that you're looking at based on healthy individuals or diseased individuals, and to what degree that makes a difference?
Yeah, so let me answer the second question first, because that's easy. So the main cohort I showed with the 250 individuals, that's a cohort of individuals that are healthy. They're individuals who about half of them have had TB, but they had TB 5 years ago. So the original study was done to identify T cell populations which are likely to predict those who get sick with TB versus not. So it was a kind of an immunoprofiling cell state association study. But the individuals are treated, cured, and they've been healthy for 5 years. So it's not really a disease cohort in that sense. In terms of the number of samples, I think for any EQTL study, you really want hundreds of samples if you can get away with it. Our feeling is the number of samples is a bigger variable for EQTL studies than the number of cells. And we've done some simulation. We have a paper which actually models these kinds of things. And basically you can get away with smaller and smaller number of cells, up to a point. Where the cells become important is if you want to find the interaction with the cell state. So in an individual sample, the more cells you have, the better you're capturing the spectrum of cell states. So I think that's how I think of the power question. So if you want to find, main effects, non-interacting effects, then I think it becomes all about, sample number. But if you want to find those interactions, then I think you really want more cells per sample.
Do you think the results would be changed if you looked at, for example, if you have interest in RA, and if you had 250 samples from patients with active disease versus 250 samples from healthy controls, to what degree do you think these subsets would be kind of disease specific, and would you be able to unmask effects that in a setting of disease that wouldn't be observed in population controls?
Yeah, I think where it matters, I mean, even more than like one of the things we're starting to do is starting to look in the tissue disease data sets, right? So T cell in the blood, I mean, it's obviously a little different in people who have disease versus not, but the T cells from inflamed tissue, those cell states really are different. The same cell states are found in both places, but it's the proportions that are really different. So T peripheral helper cells might be 20% of the T cells in the tissue, but they're 2% or 1% of the T cells in the blood. So that starts to make a difference in terms of what you're looking at, what you're modeling. It's not impossible to find it in the blood, but there's a good chance it could get swamped by other signals.
Great, thank you. Other questions? Okay, there's a question in the chat. Very nice talk. Did you have a chance to find a correlation between EQTLs and RA-specific clonally expanding T cells?
So no, not yet. So we are, we and of course many others are very interested in the topic of which T cells are clonally expanded. I think the troubling reality for us as we've looked in tissue data is when you look at T cells with identical TCRs, the clonal expansion, on CD8 T cell side is a lot, but on the CD4 T cell side, really not so much. So to me, this suggests that, we have a kind of polyantigenic and maybe less proliferative situation going on than we normally think of. So the answer is no, we haven't looked at that cell state, but we're not totally sure what that cell state should be.
Great. And I see a hand raised from Heineck. Please go ahead, unmute yourself.
Yeah, hi, very nice talk. I wonder whether the single cell analysis could tell us a little bit more about the progression of the disease in terms of, what would a patient that just started having symptoms have, which cell types would be enriched, and whether the types of RA you see might be actually reflecting some of these different stages of the disease.
So we were underpowered to answer that question. So, as I suggested, about a third of our patients were people with early RA. They had not yet been treated with a medication. We weren't able to find that there was any, broad differences between them at the tissue level and our other patients. It's a little bit tricky if you think about it. So, when we do a biopsy on our patients, like basically, you're biopsying a joint that's inflamed. So this also makes it a little bit complicated. So, maybe someone who has early RA has many more active joints. Maybe they feel really bad. Somebody with later stage RA, they're on medications, they have less active joints. But at least in our first pass, the inflammation doesn't look that different. Obviously, you're picking the worst spot to look at every time.
Thank you. Other questions? If not, I actually have another question on a slightly different topic. So, a lot of autoimmune conditions, of course, RA among others, are very strongly associated with MHC class 2 molecules, especially DRB1, DRA1. So, to what degree do you think fine mapping of these associations to kind of individual amino acid changes have provided insights in terms of very specific mechanisms of the disease? And do you think that these findings can in any way be used for therapeutic approaches?
Yeah, great question. We of course have been very interested in the amino acid fine mapping. And I think for RA, it's been, accurate. Has it been useful? That's maybe another story. I do think that it does give you some sense of what's going on, right? I mean, if it's, at the base versus the rim, I mean, it really points to kind of different kinds of mechanisms. It points to different parts of the TCR that you might be engaging with. We have an interesting study that's now in press, it's not out yet, where we show that the, HLA type of a person influences their T cell repertoire composition. And the allele that's most influential is position 13, right at the base of the DRB1. So it's not a direct interaction. It's mediated through antigen and thymic selection. Now, again, we can't quite connect it to disease yet, but I think it's a step in the right direction.
Okay, we have another question. Dow, please go ahead.
Hi, thank you for the talk. It was really good. I had two questions about the CTAPs from the phase 1. The first question is that in terms of deciding how to group the different CTAPs, was there a methodology you used to assign the different groups based on proportion of cell types?
Yeah, it was very simple. I mean, it's really nothing terribly fancy. Basically, we know for every person what percent of their cells are T cells, B cells, etc. You literally have 6 numbers per sample, and you cluster them. And the clustering we used was, boring old k-means. We did sensitivity analyses, silhouette analyses, etc. We permuted the data to get and the reason we went with k=6 is because that gave us the most signal to noise compared to permuted data. So that's how we ended up with what we had.
Thank you. And the second part of my question is, since these samples were presumably taken from biopsies, do you expect any heterogeneity from the sampling process itself to influence the CTAP's membership?
Yeah, so we don't think so. I mean, again, we don't. It's a hard question to answer. For most of these biopsies, it's not just like one chunk of tissue. It's like multiple chunks in the same sample. So it's a little different than when you look at histology, where you're really looking at one snippet. Here you've got a number of snippets of tissue that are being disaggregated together and being processed together. So it's kind of like an average across a bunch of samplings. So we were less worried about that.
Perfect. Thank you.
What we don't know is whether, one joint in a patient is very different from another joint in a patient. You wouldn't think that would be the case, but it's an experiment that needs to be done.
Do you think that single cell evaluation of kind of tapped joint will enter clinical care?
So one of the things we really like to do you don't need to actually do single cell RNA-seq, right? Because it's the number of T cells, B cells, fibroblasts, et cetera. Conventional flow can actually get you, can actually classify a patient. So the single cell tells you maybe what's going on in an aggregate of patients, but the classification actually doesn't require that much high dimensionality.
Yeah, great point. And other questions? Well, if not, I would like to thank you so much. We're right on time. It's 5 o'clock. And thank you so much for visiting us. This was really fantastic talk. Thanks a lot.
Thank you for having me.